Hello everyone. Today we will be learning about Punnett squares. But first of all, we need to answer the question, what are Punnett squares and why do we use them? A Punnett square is a type of graph that is used to measure the genetic outcome when crossing two different organisms. But remember this about Punnett squares. They only measure what could be, not what will be. There are many different types of Punnett squares. In this video, I will talk about the three main types. But before I talk about them, we need to learn about the basics of Punnett squares and how to use them. There are two main variables when you are using a Punnett square. The first one is a genotype, which are the genes that you are crossing. The second one is a phenotype, which is the outcomes of the genes. A genotype is comprised of two or more genes. These genes are represented by letters. There are two different types of genes. The first one is a dominant trait. The trait for this gene will always show up in some way, no matter what the other gene is. The second type of gene is a recessive trait. This trait will only show up when the other gene is also recessive. The first Punnett square we will be looking at is a monohybrid cross. These are a standard 4x4 grid. In this example of a Punnett square, the traits will be represented by an R. The dominant trait codes for the color red. The recessive trait will code for the color white. The genotype of the first parent is a capital R, lowercase r, making the parent red. The second parent's genotype is a lowercase r, making this one white. We input them into the Punnett square like this. Now, the thing with sexual reproduction is that parents pass on half their genes to their offspring. Remember this for later. Let's start with the top row. So we bring the uppercase R down like this, and then we do the same to the lowercase r. Do the same thing with the other parent, like this. The results show that there were two different combinations produced. The first one produced was the color red with the recessive white trait. The second one produced was a lowercase r, making this one white. Since there are four different results that can happen that each have an equal chance of happening, that means there's a 25% chance of each trait showing up. And since only two genotypes were produced, there's a 50% chance of each genotype occurring. Now let's look into a more complex Punnett square, that being a codominance Punnett square. This happens when the two traits that are being coded for are both dominant. Since a dominant trait is always expressed, the traits end up merging in a way. So let's say there's a rose with a dominant red gene and a dominant white gene. Since the traits are both dominant, they end up merging and it becomes pink. Now let's do the Punnett square. The two parents in this scenario are two pink roses with a dominant red trait and a dominant white trait each. We put it on a Punnett square like I showed earlier, like this. And now let's bring down the results. The results show that pink was the most common color at 50% of the total results. However, there are red roses and white roses because the traits are both present and ended up mixing. Now let's go to our third and final Punnett square. A dihybrid cross. This Punnett square is obviously a lot bigger because it is a 4x4 grid. The difference between a dihybrid cross and a monohybrid cross is that there are 4 genes being passed down rather than 2. We are going to use P's in this example. So the uppercase R codes for a round pea, lowercase r codes for a wrinkled pea, uppercase G codes for a green pea, and lowercase g codes for a yellow pea. So for the sake of simplicity, both parents are going to have the same genotype again, them being uppercase R, uppercase G, lowercase g. Now you'd think, oh, just insert them into the Punnett square like this. But oh wait, the results you get are very weird and don't make any sense at all. So remember how I said in sexual reproduction, each parent passes on 50% of its DNA to the offspring? Well, that is most important here because you have to list every possible outcome that the parent can give to the offspring. This results in each parent giving the traits uppercase R, uppercase G, uppercase R, lowercase g, lowercase r, uppercase g, and lowercase r, lowercase g. You have to input them into the Punnett square like this. And now when you look at the Punnett square, each offspring has a complete genome. And if I were to list all the possibilities that come out of this, it would have taken too long and this video would have come out a lot later. So I choose not to. So that is it. Done with the video. We now know about Punnett squares. Goodbye.